Hello students, today we will learn about animal cell culture, fundamental facilities and applications. So let's begin. Animal cell culture basically involves in the in vitro maintenance or in the laboratory maintenance and propagation of animal cells in a suitable nutrient media. Thus, culturing is a process of growing cells artificially. Cell culture has become an indispensable technology in various branches of life sciences. Now, let's know about the historical background. It was in 1907 Ross Harrison, the famous scientist, first developed a frog tissue culture technique. He probably chose frog for two reasons, being a cold-blooded animal and no incubation is required, and tissue regeneration uh, in is fast in frog. In 1940s, chick embryo tissue became a favorite for culture techniques. Interest in culturing human tissues started in 1950s after it was demonstrated by Hela Gay that human tumor cells could give rise to continuous cell lines. Among the various animal cell cultures, mouse cell cultures are the most commonly used in the laboratory. Now let's know about terminology in cell culture. The term tissue culture is commonly used to include both organ culture and cell culture. Now let's know about organ culture. The culture of native tissue, that is on disaggregated tissue that retains most of the in vivo historical features, is regarded as organ culture. Now let's know about cell culture. This is cell culture to the culture of depressed or disaggregated cells obtained from the original tissue or from a cell line. Now let's know about histotypic culture. The culturing of the cells for their regression to form a tissue-like structure represents histotypic culture. Now let's know about organotypic culture. This culture technique involves the recombination of different cell types to form a more defined tissue or an organ. Now let's know about primary culture. The culture produced by the freshly isolated cells or tissues taken from an organism is primary culture. These cells are heterogeneous and slow growing and represent the tissue of their origin with regard to their properties. Now let's know about cell line. The subculturing of the primary culture gives rise to cell lines. The term continuous cell lines implies the indefinite growth of the cells in the subsequent subculturing. On the other hand, finite cell lines represent the death of the cells after several subcultures. Now, let's know about facilities for animal cell culture. While designing the laboratory for animal cell culture technology, utmost care should be taken with regard to the maintenance of aseptic conditions. The facilities required with regard to infrastructure and equipment now we will discuss. There is a minimal requirement for cell culture. First is, let's know about infrastructure. In infrastructure we should know about clean and quite sterile area. Second point is preparation facilities. Third is animal house. Fourth is microbiology laboratory. Fifth is storage facility for glassware, chemicals, liquids and small equipments. Now let's know about equipments. Laminar flow sterilizer, laminar flow sterilizer, incubator, refrigerator and freezer. That is should be minus 20 degree centigrade balance. Carbon dioxide cylinder centrifuge in inverted microscope. water purifier, hemocytometer, liquid nitrogen pressure, slow cooling device for freezing cells, pipette washer, deep washing sink. Besides the basic minimal requirements we discussed, there are many more facilities that may be beneficial or useful for tissue cultures. These include air conditioned rooms, containment rooms for biohazard work, phase contrast microscope, fluorescent microscope, confocal microscope, osmometer, high capacity centrifuge, and time lapse video equipments. Now let's know about culture vessels. In the tissue culture technology, the cells attached to the surface of a vessel which serves as the substrate and grow. Hence, there is a lot of importance attached to the nature of the materials used and quality of the culture vessels. The term anchorage dependent cells is used when the cells require an attachment for their growth. On the other hand, some cells undergo transformation and become anchorage independent. Now, let's know about materials used for culture vessels. Glass. First is glass. Although glass was the original substrate used for culturing, tissues used is almost discontinued now. This is mainly because of the availability of more suitable and alternate substrates. Now let's know about disposable plastics. Synthetic plastic materials with good consistency and optical properties are now in use to provide uniform and reproducible cultures. The most commonly used plastics are polystyrene, polyvinyl chloride, and polycarbonate, Metinex, and Thermonex. Now let's know about types of culture vessels. There are the common types of culture vessels. First is multi-well plates, petri dishes, flasks, stirrer bottles. The actual choice of selecting the culture vessels depends on several factors. First is the way cells grow in culture, monolayer or suspension. Second is quantity of cells required. Third is the frequency of sampling for the desired work. Fourth is the purpose for which the cells are grown. Fifth is the cost factor. In general, for monolayer cultures, the cell yield is almost proportional to the surface area of the culture vessel. The flasks are usually employed for this purpose. Any type of culture vessel can be used to grow suspension cultures. It is necessary to slowly and continuously agitate the suspended cells in the vessels. Now let's know about the treatment of culture vessel surfaces. For improving the attachment of cells to the surfaces and for efficient growth, some devices have been developed. It is a common observation that the growth of the culture cells is better on the surface for second seedling. This is attributed to matrix coating of the surface due to the accumulation of certain compounds like collagen and fibronectin released by the cells of previous culture. There are now commercially available matrices, example Matrigel, Pronectin, Celltalk. Now let's know about feeder layers. Some of the tissue cultures require the support of metabolic products from living cells, example mouse embryo, fibroblast. In this case, the growing fibroblasts release certain products which, when fed to new cells, enhance their growth. Now, let's know about alternate substrates as substitutes of culture vessels. In recent years, certain alternatives for culture vessels have been developed. The important alternative artificial substances are microcarriers and metallic substrates. Now let's know about microcarriers. They are in bead form and are made up of collagen, gelatin, polyacrylamide and polystyrene. Microcarriers are mostly used for the propagation of anchorage dependent cells in suspension. Now let's know about metallic substrates. Certain types of cells could be successfully grown on some metallic surfaces or even on the stainless steel disc. For instance, my fibroblasts were grown on palladium. Now let's know about use of non-adhesive substrates in tissue cultures. The growth of encouraged independent cells can be carried out by plating cells on non-adhesive substrate like agarose and methyl cellulose. In this situation, as the cell growth occurs, the parent and daughter cells get immobilized and form a colony, although they are non-adhesive. Now let's know about contamination, aseptic conditions and sterilization. There are several routes of contamination in the tissue culture laboratory. These include the various materials like glassware, pipettes, equipment like incubators, refrigerators, laminar flow hood, reagents, media solutions, contaminated cell lines, and poor techniques. The routes of contamination are mostly associated with the laboratory environment and operating techniques. Now, let's know about types of microbial contamination. Several species of bacteria, yeast, fungi, molds, and mycoplasmas, besides viruses, are responsible for contamination. Major problems of contaminations are linked to the repeated recurrence of a single species. Despite utmost care taken, so laboratory can come to be totally free from contamination. It is necessary to continuously monitor for contamination and eliminate the same at the earliest. Now let's know about aseptic conditions. Maintenance of proper aseptic conditions is necessary to eliminate various contaminants due to different microorganisms and viruses. There are some measures for minimizing contamination and maintenance of aseptic condition. Let's know about the measures. First is strict adherence to standard sterile techniques and code for practice. Second is checking of reagents and media for sterility before use. Third is checking of cultures by eyes and microscopes every time they are used. Fourth is use of media and separate bottles for each line is advised. Sixth is maintenance of clean and tidy conditions at workplace. Next is personal hygiene of the staff is very important. Now let's know about sterilization. Sterilization the sterilization procedure are designed to kill the microorganisms besides destroying the spores. There are three major devices for sterilization. First is dry heat, second is moist heat that is occurs in autoclave, third is filters. The sterilization of major equipments apparatus and liquids we will discuss now. Sterilization first is dry heat. Let's know about the sterilization by dry heat. This is carried out at minimum temperature of 160 degrees centigrade for about 1 hour. Next second point is moist heat. Sterilization by moist heat. Certain fluids and perishable items can be sterilized in an autoclave at 120 degrees centigrade. 121 degrees centigrade for 15 to 20 minutes. For effective moist heat sterilization, it is necessary that the stem penetrates to all the parts of sterilizing materials. Now let's know about sterilization by filters. It is the third and final method. That is the use of filters for sterilization of liquids often becomes necessary since the constituents of these liquids may get destroyed at higher temperature, that is dry heat or moist heat. Sterilization filtration is a novel technique for heat level solutions. The size of microspores of the filtrates is 0.1 to 0.2 micrometer. Filters made from several materials are in use. These materials include nylon, cellulose nitrate, polycarbonate, polyether sulfone, and ceramics. The filters are made in different designs: disc filters, cartridge, and hollow fiber. In fact, many commercial companies, example Millipore, Durapore, supply reusable and disposable filters designed for different purposes of sterilization. Now, let's know about the advantages and limitations of tissue culture. Tissue culture technique has a wide range of applications. The most important advantages of this technique are: first is control of physicochemical environment pH, that is pH, temperature, dissolved gases, that is oxygen and carbon dioxide, osmolality. Second is regulation of physiological conditions, that is nutrient concentration, cell to cell interactions, hormone control. Third is the cultured cell lines becomes homogeneous. Example, cells are identical after one or two subcultures. This is in contrast to the heterogeneous cells of tissue samples. The homogeneous cells are highly useful for a wide range of purposes. Fourth point is it is easy to characterize cells for cytological and immunological studies. Fifth is cultured cells can be stored in liquid nitrogen for several years. Sixth is due to direct access and contact to the cells, biological studies can be carried out more conveniently. The main advantage is the low quantities of the reagents required in contrast to in vivo studies where most of the reagents are lost by distribution to various tissues and excretion. Seventh point is utility of tissue cultures will drastically reduce the use of animals for various experiments. Now let's know about limitations of tissue culture. There are several limitations of tissue culture. Let's know about the points. First point is need of expertise and technical skill for the development and regular use of tissue culture. Second is cost factor is a major limitation. Establishment of infrastructure, equipment and other facilities are expensive. Third is it is estimated that the cost of production of cells is about 10 times higher than direct use of animal cell tissues. Control of the environmental factors, pH, temperature, dissolved gases, disposal of biohazards is not easy. Fifth point is that involve cells exist in the three-dimensional geometry, while in vitro tissue culture, the propagation of cells occurs on a two-dimensional substrate. Due to this, cell-to-cell interactive characters are lost. Sixth point is the cell lines may represent one or two types of cells from the native tissue while others may go unrepresented. Seventh point is tissue culture techniques are associated with the differentiation, that is lost of the characters of the tissue cells from which they were originally isolated. Eighth point is this happens due to adaptation and selection processes while culturing. Ninth point is continuous cell lines may result in genetic instability of the cells. This may ultimately lead to heterogeneity of cells. Tenth point is the components of hemostatic in vivo regulation, that is nervous system, endocrine system, metabolic integration are lacking in vitro cultures. Addition of hormones and growth factors has been started recently. Now let's know about the applications of animal cell culture. This is a widespread concern that extensive use of animals for laboratory experiments is not morally and ethically injustifiable. Animal welfare groups world over are increasingly criticizing the use of animals. Such research workers these days prefer to utilize animal cell cultures wherever possible for various studies. The major applications of laboratory animal cell cultures now we'll discuss. First point is studies and intracellular activities, example cell cycle and differentiation metabolisms. Second point is elucidation of intracellular activities, example cell cycle and different elucidation of intercellular flux, that is hormonal receptor signal transduction. Third is studies related to cell to cell interaction, example cell adhesion and motility, metabolic cooperation. Evaluation of environmental interaction, that is cytotoxicity, mutagenesis. Studies dealing with genetics, example genetic analysis, immortalization, senescence. The laboratory production of medical or pharmaceutical compounds for wide range of applications, example vaccines, interference, hormones. There are, however, several limitations on the use of animal cell cultures. This is mostly due to the difference that exists between the in vivo and in vitro systems and the validity of the studies conducted in the laboratory. Now, let's know about the medical or pharmaceutical products of animal cell cultures. The most important application of animal cell culture is the production of a wide range of commercial compounds for medical and pharmaceutical use. The commercial production of several biological compounds by microorganisms we will discuss now. Now let's know about the production of vaccines. Monkey kidney or chick embryo cells are recently human diploid cells are in use for the production of vaccines. The vaccine manufacture in animal cell culture is rather complex with risk of contamination and safety aspect. For this reason, production of vaccines by recombinant DNA technology employing bacteria and yeast we will discuss now. Production of Let's know about the production of high value therapeutics. Many human proteins with high therapeutic potential are often in short supply. Tissue plasminogen activator, clotting factors, erythropoietin. There is a major limitation to produce human proteins that undergo post-translational modifications, glycosylation, carboxylation, etc. in bacteria and yeast. This is due to the fact that these organisms do not possess the machinery to perform post-translational changes. However, pharmaceutical proteins that do not require post-translational modifications can be produced by bacteria or yeast. That is example insulin, albumin, growth hormone. Animal cell cultures, particularly mammalian cell cultures, are useful for the production of many pharmaceutically or medically important proteins. This includes first is plasminogen interference, blood clotting factors, hormones, monoclonal antibodies, erythropoietin. Now let's know about purification of pharmaceutical products. As the desired product is produced in the cell's culture medium, its purification, isolation, and storage assumes significance. The final product for therapeutic applications is expected to satisfy some criteria. Let's know about this criteria. The product should have a stable structure with optimal activity. Second is the product should be free from other biomolecules that may interfere with its activity and cause immunological complications. It should be free from all pathogens, including viruses. Now, let's know about genetic engineering of animal cells and their applications. It is now possible to genetically modify the animal cells to introduce the genes needed for the production of a specific protein or improve the characteristic of a cell line. There are some methods to introduce foreign DNA into mammalian cells. The methods are first is electroporation, second is liquefaction, third is microinjection, fourth is fusion of mammalian cells with bacteria or virus. As the foreign DNA gets integrated into the mammalian cellular genome, the gene expresses to produce the desired protein. It is, however, necessary to select the best producing recombinant cells by conventional methods using selectable marker genes. There are some selectable markers that are used for choosing the transfected cells. Viral thymidine kinase. Second is bacterial dihydrofolate reductase. The third is bacterial neomycin phosphotransferase. It has been possible to overproduce several proteins in mammalian cells through genetic manipulation, example tissue plasminogen activator, erythropoietin, interleukin 2, interferon beta, clotting factor 8 and 9, tumor necrosis factor. The recombinant mammalian cells are conveniently used for the production of monoclonal antibodies, which have wide range of applications. Now let's know about risk in a tissue culture, laboratory and safety. There are several risks associated with tissue culture technology. Most of the accidents that occur in culture laboratories are due to negligence and cause approach while dealing with biological and radiological samples. Besides improper maintenance of laboratory, the a broad categorization of risk and contribution factors we will discuss now. First is safety regulations. Say, some of the developed countries have formulated general safety regulations to minimize the risk associated with tissue culture laboratories. First is biosafety in microbiological and biomedical laboratories. U.S. Department of Health and Human Science in 1993. Second is safe working and the prevention of infection in clinical laboratories. UK Health Service Advisory Committee in 1991. Now let's know about some general precautions for the safety of a tissue culture laboratory. First point is strict adherence to recommendations of regulatory bodies. Second is periodological meetings and discussions of local safety committees. Third is regular monitoring of laboratories. Periodical training of the personnel through seminars and workshops. Fourth point is print and make the standard operating procedures, that is in short SOPs, available to all staffs. Next is good record keeping. Limited access to the laboratory, that is only for the trained personnel and selected visitors. Appropriate disposal system for biohazards, radioactive waste, toxins and corrosive. Next is biohazards. What do you mean by biohazards? The accidents or the risk associated with the biological materials they regarded as biohazards or biological hazards. There are two main systems that contribute to the occurrence of biohazards. First is the direct sources of biological materials. Second is the of process or operations involved in their handling. Now let's know about control of biohazards. Biohazards can be controlled to a large extent by strict adherence to the regulatory guidelines and maintenance programs. Now let's know about some important aspects. Microbiological safety cabinets or biohazards wood with pathogen trap filters have been developed. Second is vertical laminar flow hood, that is instead of horizontal laminar flow hood is recently in use. This minimizes the direct exposure of the operator to the samples. Pathogen containing samples are treated in separate rooms with separate facilities. First is centrifuge incubator cell counting. Sterilization of all waste, solid glassware, etc. and their proper disposal. Facilities for change of clothing while entering and leaving the rooms. Strict adherence to the access of any personnel to the culture room. Finally, finished animal culture. Any questions regarding this, you can ask me in the comment box. For more updates, you can subscribe my channel. Thank you.